Now in this video, we are going to overview the Krebs cycle, which is also known as the citric acid cycle or the TCA cycle. Now the citric acid cycle, as the name suggests, is a cycle. And in this cycle, we are feeding in the molecule acetyl-CoA. Where is this acetyl-CoA coming from? Remember that when we think of glucose metabolism, we start off with glycolysis, where the glucose is broken down into two pyruvate molecules. Now that pyruvate is then converted into acetyl-CoA via the enzyme, the pyruvate dehydrogenase complex. Now, once we have acetyl-CoA, it's going to be fed into the Krebs cycle. Now, acetyl-CoA is a 2-carbon molecule, and it's going to combine with a 4-carbon molecule known as oxaloacetate. And oxaloacetate is part of the Krebs cycle. Now, when these two molecules come together, we're going to form a 6-carbon molecule known as citrate. And this reaction is catalyzed by the enzyme citrate synthase. Now, in a separate video, we have already gone over this mechanism, so if you're interested in more detail, go refer to that video to see how exactly oxaloacetate and acetyl-CoA come together to form citrate. Now, once we have citrate, we have to undergo an isomerization reaction, and we have an isomerase enzyme known as aconitase, which is going to catalyze this reaction. And as a result, we get the isomer, isocitrate. Now, after the isomerization reaction, now we're going to see the loss of a carbon group, and we're going to see the oxidation of isocitrate. Remember that within glucose metabolism, we are oxidizing that glucose molecule in various steps and in various mechanisms to create electron-carrying molecules that are going to be utilized within the electron transport chain. So that acetyl-CoA that we've essentially added into this TCA cycle is just being further oxidized. So we want to create more electron-carrying molecules, and those electron-carrying molecules are essentially NAD+ and FAD. Now, we have NAD+, and it's going to be reduced to NADH, so NAD+ essentially is picking up those electrons, and as a result, isocitrate is being oxidized. Whenever we think of oxidation reduction reactions, I want you to think of the enzyme dehydrogenase. And whenever you think of a dehydrogenase, think of NAD+, think of electron carrier molecules. Now, we lose a carbon group, and specifically, we're losing this carbon group over here, and we're losing it as carbon dioxide. We've got the carbon, we've got the two oxygens, and we've lost it as carbon dioxide, and in this step, we've created an NADH. Once this reaction has undergone, we form the molecule alpha-ketoglutarate, which is now a 5-carbon molecule. Remember to keep track of the number of carbons you have. We started off with 4, combined with acetyl-CoA, got a 6-carbon molecule, isomerized it. Now we've lost our first carbon. Remember, in the TCA cycle, we want to regenerate all our molecules. So we want to end up back with our oxaloacetate C4 molecule. Now we have alpha-ketoglutarate, and we're going to utilize another dehydrogenase enzyme known as alpha-ketoglutarate dehydrogenase. Now remember, I said whenever you think of a dehydrogenase, I want you to think of an electron-carrying molecule. So we have NAD+ again being reduced to NADH. And once again, we are losing a carbon dioxide. So now specifically, we will be losing this carbon group down here. Now, once we've lost that carbon group and we've lost the hydrogens, we are now going to form Succinyl-CoA. Now, in this reaction, actually what's missing is that when this carbon dioxide is being removed and NAD+ is being reduced, we are also adding in coenzyme A. Now, coenzyme A is going to come in, and now we have Succinyl-CoA. Now, Succinyl-CoA is a 4-carbon molecule, but now we have this high energy bond down here. And this high energy bond over here is actually going to lead us to produce a GTP molecule, because when this bond breaks, it's going to produce the energy to be able to create the GTP molecule, which will then be converted into ATP. So the enzyme catalyzing that reaction is succinyl-CoA synthetase. Now, succinyl-CoA synthetase, whenever you think of a synthetase, I want you to think of ATP or GTP production. So we take that GDP, we have that inorganic phosphate, we get GTP. Now that GTP is going to be converted to GDP, and then we'll get ATP as our final product. Now, and it's that this cleavage of this bond, this high energy bond, that's going to drive this reaction to produce GTP. Now we have our molecule succinate. We've lost that coenzyme A group. We've lost those carbon groups. We have a C4 molecule. We're almost getting to our oxaloacetate. We've almost regenerated oxaloacetate. Now we're going to undergo another reaction that involves a dehydrogenase. And remember, whenever you hear dehydrogenase, think of electron-carrying molecules being reduced. Now here we have FAD being reduced to FADH2. And we can see the electrons where they are lost from. Once FAD is reduced to FADH2 and succinate is oxidized, we get the molecule fumarate. Now, fumarate has this double bond over here, and we can make note that oxaloacetate over here does not have any double bonds with its carbons. So we have to reduce essentially this molecule to be able to get one step closer to oxaloacetate, and we're going to add in H2O. When we add in H2O, we lose that double bond because now we've included hydrogen and oxygen into fumarate. Now, once also this reaction was catalyzed by the enzyme fumarase. Now, once we've done so, we create the molecule malate. Now, malate is actually one step away from creating oxaloacetate. Remember, we introduced this water over here. We introduced that OH group. Well, we need to create a double bond with the carbon and the oxygen. So what are we going to do? We're going to oxidize malate. When we oxidize malate, remember, when we think of oxidation reduction reactions, what kind of enzyme do we want? We want a dehydrogenase. And we're going to utilize malate dehydrogenase. And whenever we think of a dehydrogenase, remember, you need to think of electron-carrying molecules. Here we have NAD+. Now, NAD+ is going to be reduced, and we get NADH as our product, and then we get oxaloacetate. So in this overall process, we were able to utilize the acetyl-CoA to oxidize it to get these electron-carrying molecules along with a GTP molecule, and we were able to regenerate oxaloacetate. Remember, in the TCA cycle, everything must be regenerated. Now, to give a quick overview of how much energy we yielded from this process. So from one acetyl-CoA that we fed into this cycle, we got 3 NADH. So we got NADH number 1, 2, and then 3. We got 1 FADH2 right over here, and we got 1 ATP or 1 GTP right over here. Now, what's important to remember is that acetyl-CoA, one acetyl-CoA just makes up 1/2 of a whole glucose molecule. Remember that a glucose molecule is a 6-carbon molecule, and in glycolysis, the glucose molecule is split into 2 pyruvate molecules, and remember that each pyruvate has 3 carbons. Now, the 2 pyruvate molecules from glycolysis are converted into 2 acetyl-CoA molecules. So once those 2 acetyl-CoA molecules have gone through the TCA cycle, for one glucose molecule, you're actually getting 6 NADH, 2 FADH2, and 2 ATP. So that's really important to make note of because that can be asked on exams. How much is energy generated by one glucose molecule? So, and also just to add, the TCA cycle, it occurs within the mitochondrial matrix where the acetyl-CoA is oxidized to yield electron-carrying molecules for the electron transport chain. Now remember, this is different from glycolysis. Glycolysis is actually occurring in the cytosol. So TCA, it's in the mitochondrial matrix. And these electron-carrying molecules are going to go into the ETC cycle where they're going to be utilized for substrate level phosphorylation.